So now we will continue to our first spring climb results presentation that is on almond and peach hybrids that is going to be presented from Ayzin Kuden and Iban Eduardo.
Okay, Kalimera, everyone. We will start talking about almonds and also prunus hybrids that sometimes it's applied for almond breeding or peach breeding. I will skip the introduction because if not, it's going to be a long day. And just you have an idea of what I'm going to talk about and which are what I've identified as the fourth main results regarding almond and prunus hybrids is the genetic characterization of a germplasm collection of almond, then the design of a multi-site almond ref pop collection, after the genotype-phenotype association analysis for blooming time in a white germplasm collection, and finally prunus marker assisted introgression for disease resistance. Okay, then the first that we did in this project is to try to understand the genetic variability in a collection of almond cultivars and also wild species that is composed of almost 200 accessions coming from different institutions. Wait. Okay, then I was describing the almond genetic resources that we've been analyzing with a set of microsatellites. This collection comes from 4 different institutions: INRA, CRAC, the Kukurova University in Turkey, and from here from Greece, and they are composed mainly of almonds, prunus dulcis accessions, but also there are a big number of accessions coming from wild species of almond. Okay, after analyzing them with 23 microsatellite markers, we could see that it was a clear separation between some of the wild species and the cultivated almond, and some of the main results that we can extract at this point is that, for example, prunus communis are probably feral individuals from prunus dulcis, that is the cultivated almond, that some of the species that we think that they are poor wilds, as Prunus wavis, Prunus curamica or Prunus bucarica, are probably hybrids between these species and cultivated almonds, and that Prunus turcomanica and Prunus orientalis probably are sister species. Okay, regarding the variability inside the Prunus dulcis accessions, the almond, we have seen that we could clearly separate several clusters, one of them of Asian accessions, another of the Spanish collection, another cluster including French, American and Spanish between others, and also a cluster of some individuals that you can see in this picture from the Agdamar Island, that they are completely separated from the other accessions. Okay, one of the good news is that inside all these germplasms, at least 3 different sources of resistance to Sharka have been identified, 2 in the cultivated almond accessions and 1 in Prunus fenthliana. Also, after analyzing all these accessions, some accessions coming from different islands from Greece were tried to be included in the analysis to this data set. For that, all these accessions provided by Paulina were genotyped at CRAC in Barcelona with the same set of microsatellites that INRA used to characterize the the previous collection that I've described, to try to understand if they are more similar to the Mediterranean almonds or to the Asian ones. I would like to remember that that's important because according to the history of the species, almond was domesticated in Asia and then was moving westward and it entered to the Mediterranean, and probably the almond as we know it today was originated here in Greece, or what was the old Greece, and the Romans knew this fruit as the Greek nut. Okay, then perhaps these accessions located in this part of the world could be the origin of the species through all the other Mediterranean countries. Okay, that's the preliminary results that we find here. I have to say that aligning two data sets of microsatellites is not easy. And then at this point, you can see here, I don't know if you can see the mouse, no? But anyway, these are the gray ones are the Greek accessions, okay? And they are clustering together with two wild species that are Prunus orientalis and Prunus turcomanica. Okay, at this point we have two hypotheses. Or we did something wrong during the alignment of the two data sets, and then all these Greek accessions, they are together in this same cluster, or it would fit with the movement and the domestication of the species. Then, yeah, we have these two hypotheses, but yeah, I would like to recheck all the data to take a more clear conclusion, if possible, before the end of the project. Okay, all this material has also been characterized for different traits, including phenological traits as flowering date, leaf bud break date, leaf fall date, also susceptibility to some diseases, and some of them also to drought tolerance through in vitro methodologies. The idea with all this material is also, inside the context of this project, is try to design a multi-site almond RefPop collection that is basically based on the idea of the peach and apple RefPop population. These are two collections of germplasm that have been selected based on the genetic structure of big collections of these species, but also including
recording in progress.
Sorry that I cannot remove it from here. Okay. Yeah, but yeah, that's the thing.
This is what I would like to do.
Okay, thank you. Okay, the idea of the multi-site core collections is to analyze genetically a big collection of accessions, obtain these ones representing the most of the genetic variability, but also include accessions based on the importance of some specific phenotypes or some other characteristics that are important and are consulted to stakeholders or people that really know the different crops. Okay, at this point we have all the genotypic information for the almond RefPop, and then we can select all these accessions representing the most of the genetic variability. And now we have to further define the experimental design and the scope of the almond RefPop, what we want to use it for. We also identified at least 3 partners that are interested in having a copy of these collections. And we have to contact almond breeders and germplasm curators to decide which plant material should be included according to the phenotypic data, or also check if there are some important almond cultivars that they are not included in this data set but would be interesting to add, or also to decide until what extent we should add wild species into the collection. Okay. Finally, we have to deal with all the plant protection requirements to move materials between countries, especially from countries outside the European Union. And finally, find a nursery to propagate all the materials and share it with the different partners. Okay, the other task that we did was the genetic characterization of two almond collections from INRA and from IRTA. These ones, they have been genotyped with a new tool developed that is a big chip of 65,000 SNPs, basically to understand the genetic structure and to do a GWAS analysis association between phenotypes and genotypes. Also, colleagues from INRA, they genotype a population, interspecific populations of almond, Ferragnés to Ferrochar, to try to dissect the genetics of the Fusicoccus amygdalae resistance. Okay, at this point they are still working to improve the phenotyping protocol. Some of the main results here is that we have at least 80 accessions of almond that correspond to one of the pure clusters that correspond to the origin of each of these accessions, to the Americans, Chinese, French, Spanish or Italian accessions, and then we have 72 other cultivars that they correspond to an admixture groups of these previous ones. The preliminary analysis for the association for blooming date, we already identified a SNP that was linked to that trait that it's located on chromosome 2, but we are at this point further analyzing all this data. I'm moving to interspecific genetic resources. In this project, two partners, Spanish researchers and researchers from INRA, have worked in two different disease resistance. One is peach powdery mildew, and the other is Sharka or PPB. Okay, in Barcelona, we have been working with different interspecific species between populations between peach and almond, and between peach and Prunus davidiana, that is a wild peach. And until now, the most interesting findings is two new sources of powdery mildew resistance have been identified, and that can be incorporated into breeding programs. Later on, I will summarize how we can do that. For the Sharka resistance, our colleagues in INRA, they have developed two interspecific populations derived from the almond delphiid that is resistant to PPB, and a cultivar of peach. These populations have been genotyped with different markers and have been phenotyped for PPB resistance, and during 2021 two QTLs have been identified, one in group 3 and one in group 8, and in 2022 these QTLs were verified and were identified in the same chromosomes in the same regions. Okay, then at this point the situation is that we identified several genomic regions linked to different resistance, and now we have to introgress them in allied materials. Okay, some years ago in our lab we developed a strategy that we call the marker-assisted introgression, that is basically based on trying to develop cultivars or accessions. I don't see the mouse here. Yeah, through backcross and marker-assisted selection, just to identify individuals with a unique introgression containing the region of interest where our resistant genes have been identified, and remove all the other alleles coming from the exotic species, and they can harbor undesired traits. Okay, here you have the general scheme of how to proceed with this strategy. At this point, for peach and almond crosses, we already developed two populations of what we call the introgression lines. One collection having introgressions of almond in heterozygosis in a complete peach genetic background, and the other collection that also has a unique introgression, but in this case is in homozygosis. Okay, This material is very interesting for studying quantitative genetic traits, or also can be used directly as pre-breeding material to introgress them in different plant breeding programs. Okay, for example, we know that the plants, this mouse, can you see it? For example, the plants having the region containing the gene defining the red flesh or the peach powdery mildew resistance can be directly crossed with light cultivars to easily introgress these traits into a breeding program. Okay, at this point we also develop what collection of what we call a pre-Niels collection, because still they have 2 or 3 introgressions coming from Prunus davidiana, and also in this collection we identified a new gene for peach powdery mildew resistance, and also for other interesting traits as the green skin when the fruit is already ripe, and we are trying to check if this trait can have also some correlation with some kind of resistance or tolerance to monilinear. Okay, then the expected outputs for almond in the project is an almond germ plus collection characterized genetically and phenotypically, mainly for flowering time. The proposal for a multi-site almond germ plus collection, the future almond RefPop, molecular markers for flowering time and other interesting traits that could derive from the GWAS analysis, and new phenotyping tools, specifically the test for Fusicoccus resistance. Okay, regarding the Prunus hybrids, the outputs would be a list of individuals presenting resistance to peach powdery mildew and PPB that can be used as pre-breeding material, and also molecular markers linked to these traits for its use in marker-assisted breeding. And I would like to thank all the partners involved in this species.